Welcome to Roadmap to Residency Evidence-Based Applications. So in this video, we will be talking about bacterial shapes, arrangement, and different staining patterns. So for the identification of bacteria, you need to look for the shape, the color after staining, and there are some special tests for the identification of bacteria, like the coagulase, catalase test, and all. So bacterial shape, there may be a coccus, which is sphere shape here. There may be rod shape like bacilli, and there may be coccobacillus, which may be oval shape. Now here you can see now this is a single cocci. This is a diplococci. This is a cocci in chain, and this is a cocci in cluster. Okay, you can see the streptococcus in chain. You can see the staphylococcus in clusters. Now, cocci, so most cocci are gram-positive. Example, streptococcus, staphylococcus. There are very few gram-negative bugs which are cocci. Example, Neisseria, meningitis, gonorrhea, and Moraxella catarrhalis. So rods. So the most rods and coccobacillus are gram-negative. There are few gram-positive rods too, like Corynebacterium diphtheriae, Clostridium, Listeria, and Bacillus, like anthrax and Cereus. So there are some other shapes of bacteria, like branching filamentous bacteria, which can resemble fungi and are seen in case of actinomyces and nocardia species. Spirochetes, which can be treponema, syphilis, borrelia, Lyme disease, leptospirosis. So there can be some common bacteria called as Vibrio cholerae. Leomorphic bacteria takes on many shapes like rickettsia, chlamydia, uh, and all. There are some common bacterial stains like Gram stain, Giemsa stain, Gill Nielsen stain, silver stain, and India ink stain, which is particularly used for Cryptococcus, which is a fungi. So simple stains, methylene blue, safranin, and crystal violet are simple stains which are added to fix the preparation of bacteria, and they can be washed away later. The stain remains behind to show the bacteria as a background, and they are used to see number of bacteria and their shapes. You can see the ground stain, the feature that I have shown you in the previous video. Positive is purple, negative is pink. So how do you stain? So first of all, there is fixation of the bacteria to the glass slide. After that, you add a crystal violet, and then you add iodine, and then you again decolorize with alcohol and all. Now, what happens is that the gram-positive bacteria, they retain the stain. So after the decolorization, the stain is not washed out. And when then you counter stain the bacteria with safranin, in case of gram-positive, since it already has the stain, it will not take up safranin. But since in case of gram-negative, the stain has already washed out, then it will retain the safranin stain, which is pink in color. So purple is gram-positive, retain crystal violet in cell wall. Red is gram-negative, which do not retain crystal violet in cell wall, and they take up safranin counterstain. Thick cell wall of peptidoglycan is in gram-positive bacteria makes them purple. So gram stain limitation. So some bugs do not gram stain well. For example, in case of treponema or syphilis, which is because they are too thin to see. In case of mycobacteria, because of the mycolic acids in the cell wall. In case of mycoplasma, because there is no cell wall. In case of intracellular bacteria, with the rickettsia, chlamydia, legionella, obligate intracellular, chlamydia, obligate intracellular, no muramic acid cell wall in case of chlamydia, and legionella, which is mostly intracellular. Gemcitabine, which is a mixture of methylene blue, eosine, and azure B. It is used to look for the plasmodium in case of malaria, and they enter the cell and stain the nucleic acid of the organism. They are used for blood smears and marrow smears. It is used to see the protozoa like plasmodium and trypanosomas, and some intracellular organisms like chlamydia, rickettsia, and borrelia. Geonisal stain, also called as the acid fast stain. It contains carbofuscin, which is used to detect mycobacterium and also used for nocardia species. The acid-fast bugs resist the decolorization with acid solvents. So silver stain should have a special stain for three organisms: Pneumocystis pneumonia, Legionella, and S. pylori. So pneumocystis pneumonia causes fungal infection, and they can also cause diffuse interstitial pneumonia in case of HIV/AIDS patient. Legionella can also cause pneumonia, and they contaminate the water. That's how it causes outbreak in the nursing homes. H. pylori can cause gastric ulcers, as you all know. So India ink is a negative stain. in which the background is stained but not the organism. The unstained organisms stand out in the contrast, and they are primarily used for to coccus neoformans, which have large polysaccharide capsule and they create halos. Like here, you can see the halo. There are some specific pigments for some bacteria. So some bacteria produce special colors. For example, Staph aureus, which produces golden yellow color. Pseudomonas aeruginosa, which produces blue-green pigment, which is called as pyocyanin. And Serratia, which can produce red pigment. Actinomyces is a filamentous bacteria that cements together. The colonies have yellow-orange appearance, and then they are known as sulfur granules. Here you can see the different shapes of, sorry, the different colors the bacteria have produced. Thank you for watching. Keep studying.